Hello everyone, my name is Zhiyuan Wang and I am from Professor He's lab. So today I will give a small presentation of maybe my future 5 years PhD research topic, which is the iPSC modeling of the brain. So to do this, I think I will do this mainly in two steps. The first one is a good blood-brain barrier model, and second one is a good hypothalamus model. So for first, blood-brain barrier models, or BBB. BBB actually is a very important barrier inside our brain or inside our body. Actually, maybe the most important one inside our body because it separated our brain central nervous systems with the blood. It is also the major place for our central nervous systems and the blood to exchange molecules. It has a great impact on the nutrient and waste exchange like the glucose or other neuron toxic molecules. And also, most people do not think our brains has an immune systems. Although our brain has some kind of cells like microglia cells, they are not considered whole immune systems. So the blood-brain barrier also have a great impact on protecting our brains from the pathogen. And also, it can impede the drug delivery, so that if you want to design the nanoparticle which can penetrate, which you want to bring it into the brain, you have to take the blood-brain barrier into consideration. And of course, it is highly related with the neurodegenerative diseases like Alzheimer's or Parkinson's diseases. That means that for the patient in the Alzheimer's or Parkinson's diseases, their blood-brain barrier is a little bit more leaky than ours. So the current model for the BBB barrier, actually there are three kind of models are mostly used. The first one is organoids, and second one is bioprinting, third one is microfluidics. So for organoids, as we can see, it is a very good model because it's highly resembled in vivo development. Actually, it is just like embryo. It started from a small cluster of cells and finally fully grow into a fully functional like subregion of the brain. And it is in 3D so that it can have the cell-cell interactions or the shear stress, which is very important properties. So that we can control the differentiation. However, it is a little bit hard to control the overall differentiation of the cells because we don't know which cells to be in which place. When we are adding cells, they are actually just a random mix of the cells. We cannot control that, so that it causes a big problem. And the second one is bioprinting. Compared to organoids, it is highly precise and reproducible thanks to the 3D printing technology. But the problem is that mostly it is only a 2D sheet because for 3D printers we have to first print an ECM on the substrate and we print our bioinks, mostly the iPSC on these sheets. Although there are methods to do this, for example like stacking layers together onto each other, like the multi-layered inkjet printing, this causes another problem, is the ink. The ink we are using are mostly iPSC. But for iPSC, it is very fragile when we are trying to separate them. But for bulk printing, the 3D printer has to get the iPSC separated so that it can print them. This will be a big problem. And the final one for the microfluidics. So for microfluidics, it is a very precise and reproducible model because we can design our microfluidic channels to allow our flows to let the liquid to flow in, which is very highly desirable or designable. We can just try to do whatever we want. And between the different channels, we can have the hydrogels to support our cells to grow in a 3D version so that it will also have the cell-cell interactions or shear stress, which we can also control the blood vessel diameters, which is also a very important property. However, the biggest problem is that for microfluidics, it's a little bit hard to fabricate. But in Professor He's lab, we have many experts to do this, so I don't think it will be a really big problem. So second one, cell types. Actually, for most research for BBB models, there are mostly two types. One is HIPSC, and the second is primary neuron cells or neuron stem cells. Why I do not take the human embryo stem cells into consideration. Why? Because actually they do not provide more advantages than HIPSC, but they cause more severe problems, the insect problems. So first for HIPSC, since it came from the specific individual, so it is low risk of immune rejections or actually no interspecies differences, all from human. And second, it's relatively easy to obtain compared to HESC, because for embryo stem cells, you have to kill the embryo to get the stem cell. It is, not that ethical. But the biggest problem is that in differentiation. It will cause even the experienced groups to spend several months on the HIPSC to fully differentiate them into the target cells, like the midbrain dopaminergic cells for Parkinson's disease. However, for primary neuron cells or neural stem cells, this kind of problem does not exist. They are either in the target cells like the MDA neurons, which I mentioned, or like they are in astrocytes. Or it's really easy to differentiate them because they are already in the neural stem cells. It's much more easier. But the biggest problem is the immune rejection or the interspecies differences. Since the primary cells are mostly from the mouse or rats, the interspecies differences, or they are from patients, the immune rejection, and this cannot be modeling fully, like the healthy individual. So if I can have a good model of the BBB model, I think I will do the hypothalamus. Hypothalamus. Why I think I will do hypothalamus? Because hypothalamus is a very important region inside our brain. It controls the release of hormone, so many diseases can be related to the hypothalamus dysfunction. And also it controls many important body functions, like the body temperature or the sleep, awake circle, or the feeding circle, so that it can be highly related with the diabetes. And what's more, it's not that well studied. Actually, there is no in virtual models of the hypothalamus. But for me, if I can build a great BBB model, I think I can change the neurons into hypothalamus subregion specific neurons. It will be very interesting. And and what's more, for hypothalamus models, it can even allow me to establish a more complex model with other organs, like the pictures which I have shown here, which it is from recently Nature Biomedical Engineering articles, which it combined its brain chip with the liver chip to achieve the certain kind of virus effect. They have their brain mimic part here and the liver mimic part here. So if they can do this, I think it's highly possible for me to do this as well. I can have the hypothalamus mimic part here, and like here maybe a pancreas mimic part to study the release of the insulin. So that's all. Thank you. And here is the page for reference of all pictures in my presentation. Thank you.